Good day, ma'am. Our lesson entitled Midterm Laboratory Exercise DNA Analysis. Before we proceed to our lesson, let's start with the lesson objectives. Students should be able to explain the importance of DNA analysis and enumerate the steps in DNA analysis. And these are the contents. Number 1, title, objectives, slide contents, presenter's identity. And the number 2, description of DNA, location, composition, and nature. Number 3, description of DNA analysis and how it is used to solve crimes. And number 4, 4 steps in DNA analysis. And number 5, 4 types of DNA evidence analysis. Now, I will discuss that deoxyribonucleic acid, or what we call DNA, a self-replicating material which is present in nearly all living organisms. As the main constituent of chromosomes, it is the carrier of genetic information. DNA is made up of molecules called nucleotides. Its nucleotides contain a phosphate group of sugar group and a nitrogen base. The 4 types of nitrogen base are adenine, thymine, guanine, and cytosine. The order of these bases for which they form DNA instructions or genetic code. Forensic DNA analysis is the use of DNA specimens. In legal proceeding, just a people can leave fingerprints when they touch a surface. So, too can they leave biological material that contains DNA. DNA is generally used to solve crimes in one of the 2 ways. Number 1, in cases where a suspect is identified, a sample of that person DNA can be compared to evidence from the crime and the scene. The result of this comparison may help establish whether the suspect committed the crime. And the number 2, in case where a suspect has not been identified, biological evidence from the crime and the scene can be analyzed and compared to offenders' properties in DNA database to help identify the perpetrator. And for now, these are the steps in DNA analysis. Several basic steps are performed during DNA testing, regardless of the type of testing being done. The general procedure includes: number 1, the isolation of DNA from an evidence sample containing DNA of uncertain origin, and generally at a later time, the isolation of DNA from a sample. Number 2, the processing of the DNA so that the test results may be obtained. And the number 3, determination of the bionation in the DNA, the result from specific regions of the DNA. And the number 4, the comparison and interpretation of the test results from the unknown and known samples to determine whether the known individual is not a source of the DNA or is included as a possible source of the DNA. And now for the last slide, types of DNA evidence analysis. Polymerase chain reaction, the copying process known as polymerase chain. Reaction uses an enzyme to replicate DNA regions in a test tubes by repeating the copying process. A small number of DNA molecules can be reliably increased up to billions within several hours. Number 2, short tandem repeat is a forensic analysis that evaluates specific region, loci, that are found on nuclear DNA. The variable polymorphic nature of the STR regions that are analyzed for forensic testing. And the Y chromosomes. Markers target only male fraction of a biological sample. This technique can be very valuable if the laboratory detects complex mixture within a biological evidence sample, because Y chromosome is transmitted directly from a father to all of his sons. It can also trace family relationship among males. And the number 2 is the mitochondrial DNA. This analysis allows forensic laboratories to develop DNA profiles from evidence that may not be suitable for STR analysis. Mitochondrial DNA technology analyzes DNA found in a different part of the cell, the mitochondrion, such as hair, shafts, bones, and teeth. And that's for all for midterm laboratory exercise DNA analysis. Thank you.